connection to all this, of course, is meant to be gut health and connecting microbiome to some of the challenges that John described that are facing the industry in terms of enteric disease. If you're a graduate student that has been working in this field or studying in this field in the last 10 years, you've certainly been asked to define gut health in your comprehensive exam or in your defense at some point. And the reason is the definitions are not that great. We struggle to understand what gut health is and define gut health in terms of a community. I always come back to the role of the gut. And we talk lots about the role of the gut in terms of its barrier function, its need to serve as a barrier to pathogens that might enter the gastrointestinal tract. But in terms of the many nutritionists in the room, the other side of that story is that this gut has to function in terms of efficient assimilation, digestion, and assimilation of nutrients. And those nutrients need to be used efficiently, hopefully to support lean tissue growth. And so from a perspective of gut health is your gut functioning, is the pig's gut functioning and equally performing these tasks? What we've learned from Emma and others is that it's probably the gut microbiome that is driving these outcomes, these functional outcomes, whether that's barrier function or nutrient assimilation. It's the microbiome, the composition of that microbiome, the products of fermentation, the metabolites that microbiome is producing that, in my tenet at least, are driving gut health, are driving those two primary functions. And if we look at what factors are directing the composition of the microbiome and the activities of the microbiome, of course, age, diet, environment, genotype, these are the major drivers that are affecting what's present in our gut, what the microbes are, what the metabolites are, and then their impact on the host in terms of gut function. Emma's already given us a detailed map of the gastrointestinal microbiome and given us a number of key factors or descriptions of this microbiome. I tell my students abundant with diverse, redundant, and dynamic members. It sounds like a superhero squad or something like that, doesn't it, in terms of those descriptors. And the fact is that, something in the several hundred species range, we talked about, there's some controversy about what the total number of species are. Probably some controversy around the total number of genes, but if you think about 2 million genes versus something less than 30,000 genes in your own genome, the ability of this diverse community to produce metabolites is huge. You know about short-chain fatty acids, perhaps butyrate, acetate, propionate. You know about ammonia and a few others. But the potential for metabolite production by this community is tremendous. And the problem is that it's dynamic, right? This is a figure simply showing the small intestine and the colon being very different communities. But again, we know that the composition of the microbiome changes with age and diet, and et cetera. And in fact, I think that's probably what I have next here. A number of major factors. We know when the pig, age is a big one, right? As Emma said, pigs also are born germ-free and in that early life are inoculated with an early microbiome that increases in diversity with age. Diet. In terms of ingredients, when you're feeding corn or you're feeding wheat, you're not just feeding the pig, but the components of that diet are also substrates for microbial fermentation. And if you consider that some species have the enzymes that work great on corn and other species have enzymes that work great on wheat, arabinoxylans, just that simple selection of corn versus wheat will impact the composition of your microbiome. Whether that has significant functional outcomes or not, we don't really know, but we certainly have lots of work to demonstrate that ingredient selection has dramatic impacts on what's present in your gut. Environment is the other major important factor. Two pigs or pigs, litter mates, non-litter mates in the same pen will look more alike than litter mates in different pens. So the environment in which they grow up in is much more important than who their mom was. Interesting. Not to say that mom doesn't contribute. We certainly believe that the sow, as a piglet passes through the vaginal canal and as she's exposed to that fecal material, is a major source of the initial inoculants of the pigs. And there's some good work, recent work, cross-fostering studies between Meishan and Yorkshire pigs, for example, that have demonstrated some of that. But it's a relatively minor contributor still to looking at, with age, they get more and more diverse. Weaning is a trough. Diet will influence what's growing there. And mom is a bit of a secondary factor. That may not negate her importance in terms of supplying some of those keystone bacteria, foundational bacteria that Emma described. But in terms of the total abundance, a mom may not be as big a role player as the others. As I said, gut health is about microbial, is not only about the composition of these organisms that are existing in the tube that is your gastrointestinal tract, but there is obvious and real crosstalk, that these communities are recognized. So this is meant to be, and there's more detail here than we need to talk about, but this is meant to be that single cell layer of your gastrointestinal tract that lines the villi, that separates the inside of your gut here from the rest of you, the inside of your pig's gut from the rest of you. The light blue and the dark blue are meant to be different layers of mucus that are in part serving to help hold some of these communities in the lumen and restrict access to the epithelium. Yet, of course, they do get access to the direct epithelium. And of course, as John has described, the E. coli are attaching directly to these enterocytes through fimbriae and having a direct impact. Now, this is my favorite. The cartoonist probably took a little liberty here, but this is one of my favorite cells, dendritic cells, specialized cells that are existing just below the epithelium, reaching all the way out to the lumen of the gastrointestinal tract and sampling bacteria and antigens that are present in the middle of the digestive tract. My point from all of this is that there are a number of pathways with which the host recognizes changes in composition in the gastrointestinal tract and responds in respect to changing its secretion of mucins, its secretion level of mucins, changing its secretion of antimicrobial peptides, changing the rate at which these cells are replaced so that damaged cells or necrotic cells can be sloughed off and new cells replaced. So there are many, many pathways that allow the host to communicate, and it goes in both directions, but to communicate with the organisms that are present in the gastrointestinal tract. As I said, there's a tremendous amount, 2 million genes means there's a tremendous amount of genetic capacity within the microbiome, producing a plethora of different compounds. You're very familiar with butyrate. We used to think that butyrate as a short-chain fatty acid was simply a source of energy, a way for the pig to get some energy from fiber. Well, it turns out butyrate, of course, is soluble. It actually enters the cells, as many of these metabolites of fermentation will. And there are actually receptors for butyrate. So that some of these effects that we see in terms of butyrate having a positive effect on the barrier function, the ability of the pig to protect against disease, are actually receptor mediated. Butyrate binds a receptor just like a hormone binds a receptor and stimulates some of these outcomes. Stimulates hormones like GLP-2, for example, that might drive intestinal cell proliferation and replacement of those damaged or dying enterocytes before a hole forms and makes the animal susceptible to a pathogen that might be present in the environment or an opportunistic pathogen that might be present in the environment. So, butyrate again is one of an example, and I just throw this slide up to scare the crap out of you, and to in the crap bit of you is there was no pun intended. But to recognize, again, these 2 million genes, this is the metatranscriptome. So these are an attempt to sequence the genes that are turned on. Happens to be a mouse study, so it's not really relevant to the pig, but it is relevant from the point of view, these are all the genes that are turned on, and these are the various metabolic pathways. The size of these dots are bigger because they're more active, or potentially more active. There's more genes involved. The potential for different compounds from this process is immense. Emma mentioned one that they've identified, but the numbers and potential is huge, and we have only begun to scratch the surface in terms of some of these compounds, some of which may be produced in relatively small amounts, that may interact directly with host receptors, mimic our own signaling peptides, signaling molecules, or simply sit on in the active site of an enzyme and turn down and by doing so prevent its activity from proceeding. So I think there's a big black box here in terms of the metabolites of microbial synthesis and their impact in terms of barrier function and nutrient assimilation. Probably not helping you in terms of trying to I'm going to flip through these. And the only reason I throw this slide in here is to reiterate that not only can the host sense all of these metabolites that are generated, but the host has a number of pathways that I've just simply labeled here, where it can actually sense the commensal bacteria. Friends, non-pathogens are also recognized by the host. There are a number of pathways, again, that allow the host to see and respond to probiotics, but other commensal organisms that are present in the gastrointestinal tract. So just like a pathogen might have a significant impact by being recognized by the immune system, there are pathways that reflect non-pathogens' exposure to immune development. And again, I take your thoughts back to the fact that the gastrointestinal tract is not just a barrier, it's also the organ that we rely on for nutrient digestion and absorption, and that these roles are interdependent, right? That, barrier function, mucin secretion, antimicrobial peptides, cells infiltrating the lamina propria here, just below the surface of these villi, ready to pounce on any organism that manages to get across the epithelium and hopefully phagocytize or kill it. Other cells secreting antibodies into the lumen to prevent those pathogens from even sticking to the intestine in the first place, in terms of IgA secretion. Epithelial cell renewal, if these cells are being damaged by the pathogen, or they're even irreparably damaged, not necrotic, but they've been in this environment, this tough, rough and tough environment long enough that it doesn't pay to repair them anymore, let's dump them by a process, programmed cell death of apoptosis, and get rid of them and replace them with new ones. All of these are activities that the gut employs when it's experiencing a new pathogen, is when experiencing an organism that it is sensed as having as infective and has the potential to get across this and cause damage. These things are not independent of digestion and absorption. These epithelial cells are the cells that are responsible for digestive enzymes, that are responsible for nutrient transporters. If you are getting rid of your old ones more rapidly, if you're replacing them with new ones that aren't fully mature, the capacity for digestion and absorption can be reduced. And further, once you do digest and absorb and assimilate those nutrients, are they going to be used for lean tissue accretion, or will they be used for these processes? Mucus is probably the classic one, mucus and threonine. If a pig is experiencing factors in the gastrointestinal tract that are increasing its inflammatory response, resulting in an increase in mucus secretion, that threonine is going to go to mucus secretion before it's going to go to lean tissue accretion. And you'll need to increase your threonine content in order to maintain lean tissue accretion in that kind of scenario. So here it is. Here's my, some of you have seen this before, perhaps from me, but this is my model of gut health and how I think about gut health and therapies that might utilize the microbiome as a means of improving gut health. Quite a long time ago, or maybe not all that long ago, when I was young and foolish, and Ben was around to believe me, I guess, I played with germ-free animals, with germ-free pigs. You know, these were, Emma talked about the negative effects of the germ-free environment in colonies of mice so that over generations are maintained germ-free. I can tell you a germ-free pig that's taken from a sow and raised in an isolated facility is a pretty happy and actively growing pig. And so what does that pig look like? That pig has very high digestion and absorptive capacity. There's never been any bacteria or antigens driving epithelial cell replacement in the gastrointestinal tract. If you look at the expression of absorptive enzymes, they're very high in these pigs because there's nothing driving replacement of epithelial cells. All the cells are mature and fully functioning. So they have high digestive absorptive capacity, as long as, of course, you feed them a highly available diet. You're not relying on fiber to feed them. They're not going to have any fermentative capacity. But they have extremely low barrier function. Again, they've never experienced a microbiome. There's been no activation of the immune response. That's why these villi are so long and populated with a large number of cells. There are no immune cells in the lamina propria here. Ready to, and in fact, there's damn near no immune cells in the lamina propria in these pigs. They're not secreting IgA into the lumen. They're not, there's no phagocytic cells here, or very few, because again, there's been no activation of the immune protective mechanisms in these pigs. I say they have low nutrient requirements. They have low nutrient requirements because none of these activities are drawing nutrients from lean tissue accretion. They're not spending any effort on mucus secretion or cell proliferation or cell replacement because they don't need to. So that sort of, that contrasts our clinically sick pig, right, that's experiencing a pathogen inflammation. Poor digestive capacity. So here the villi in these pigs are the short, squat villi because the lamina propria is now full of cells that are actively phagocytizing, hopefully the translocating organism, secreting IgA. We have high levels of mucin being secreted. We've lost lots of epithelium because the cells have been destroyed. And the animal is rapidly trying to replace them by increasing proliferation. These guys have poor digestive capacity because they've lost villus, and they have high nutrient requirements because, again, they're producing mucus, they're producing cells, etc. This guy in the middle are my sweet spot pigs. That's the pig that I think you need to aim for in your systems. The pig that, and I think microbiome is driving this sliding sweet spot, if you will, from the germ-free environment in terms of exposure to and somehow ideal set, and I say somehow because we don't know yet, but an ideal set of microorganisms that drive the development of an intestine that has what I will call physiological inflammation, right? It's secreting sufficient mucus. It has an adequate number of cells ready to respond to organisms that cross the epithelial barrier. It's producing IgA. The question is, where do you need to be on this scale, right? What is the potential for exposure to pathogen? Because the farther you are this way on this scale, the more it's going to cost you probably in terms of appetite, but also in terms of nutrient density, particularly amino acid density. Because the more you go here, the more maintenance requirements that will increase. The question is, how much do you want how much do you want to push in terms of manipulation of the microbiome, the pigs this way, so that they are more capable of responding to a pathogen should it enter the environment? You have to think prophylactic and not treatment, I think, in terms of microbiome. This is about preparing pigs so that if they're exposed to a natural infection, they have a better chance of clearing that infection. Fewer pigs will get sick, or hopefully they won't get sick at all. But if you hit them, if they get hit with a major pathogen exposure, this approach is probably not going to save you, right? We're talking about prophylactic treatment to minimize the impact. As I said, I think the microbiome plays a critical role in finding that sweet spot. And there are two major considerations here, I think, or two major mechanisms that are driving that. One is, as Emma described, a more diverse community means that these members are occupying every niche and they're actively competing with each other for that space. The more diverse, the more highly ideal each individual species is for that site, the more capable they are of defending that site against the intrusion of a pathogen. And the other side, of course, is that this microbiome is constantly talking to the host and driving those innate barrier mechanisms in order to protect against infections. Unfortunately, I don't have the answer for you in terms of what the ideal microbiome is. It comes back to this superhero kind of descriptors, right, in terms of complexity and dynamicism, etc. We're not at a place yet where we know what that microbiome looks like. We have some clues, of course. I wanted to briefly talk to about, this is microbial competition in a 2x2 setting. A common probiotic here against some lactobacilli, Bacteroides, Enterococcus, E. coli, Clostridia. In these simple 2x2 settings, the taller the bar here, the greater the negative interaction between these. So bacillus growing on a plate will inhibit the growth of these organisms and it won't touch these organisms very much. But again, that's in a 2x2 setting. What happens in a 1x1 setting? What happens in a 1x500 species setting? Do these kinds of relationships continue to exist? It's a starting place. Dan Columbus and I, a student just completed, Michael Wellington just finished his thesis on Friday. In terms of an example of crosstalk, and think about fiber. Fiber, as we know, drives mucin secretion. It changes the microbiome. Probably one of the big drivers is short-chain fatty acids, increasing mucin secretion, and we were able to demonstrate that here and looking at mucin secretion in feces. What's interesting, and this is in just one study, but what's interesting is when these pigs are challenged with LPS, high fiber actually protected against the loss of barrier function associated with LPS challenge. So as I was speaking to you earlier, and as John had mentioned, stressors make pigs more susceptible, right? Increased susceptibility. Stressors, like some other impact, reduces barrier function. Is your pig ready to respond to that stressor and perhaps get past the stressor, or is it going to get sick? Maybe fiber in this mechanism is in part contributing to some of that protection by increasing mucin secretion and increasing barrier function in pigs. So an example of the discussion, the crosstalk between the microbiome, the products it generates, and barrier function outcomes. Can we, this whole idea of gut health ultimately links to performance, right? Growth rate, average daily gain, that's what pays the bills. That's what you're looking for when someone tries to come and sell you a new product. Can we link microbiome to, and specific members of microbiome to growth performance? There are some studies, relatively controlled studies, small numbers of animals. This happens to be one with just 30 animals in it. but that are starting to, this one is showing that post-weaning Prevotella at day 36, abundance is associated with a higher body weight and a tendency to increase growth rate in, again, this small group of pigs. There are some other more recent papers that are looking at feed efficiency and seeing some connections between microbiome and feed efficiency. Are we seeing the same bugs yet in all papers? No. But Prevotella is certainly an interesting one in that it has been identified in the past as a potentially positive organism. You'll see later this week, John and Ben Willing talk about the resilient pig. The focus there is going to be on, of course, the genetics component, but Ben's going to talk about the microbiome component as well. Very interesting because that natural challenge model also includes viruses. And there's one thing that we know very little about is the relationship between microbiome and viral infection. And so I encourage you to pay some attention there and see what Ben has to say tomorrow with respect to the impact of that resilience model with respect to the microbiome. Little plug there, Ben. Again, I want to iterate, this is prophylactic. When we talk about microbiome, we talk about antibiotic replacers. Remember, we're not talking about therapeutic antibiotic replacers, we're talking about prophylactic antibiotic replacers. You're talking about moving your pigs to a place where they're better able to withstand the next stress. You're not going to be able to bring a pig, I think, to a place where it will be protected against an overt challenge with a pathogen. The good news is that in terms of microbiome therapy, I think you're already using, John talked about biosecurity, sanitation. You're already using a lot of strategies that are actually microbiome related. Those are keeping out pathogens, right? That's affecting the microbiome of the pig. Diet formulation, my nice example there is lactose. Why do you include lactose in the early post-weaning diet? Awfully expensive ingredient. Yes, because it's a source of nutrients that the pig is familiar with and can digest. It's also a substrate for lactobacilli and will probably is a big player in terms of maintaining lactobacilli populations in that early post-weaning period. New directions, there's lots of gut modifier products, probiotics, prebiotics, phytochemicals. Emma's talked about a number of these. I think we have to look for, as you have these guys come knocking on the door, improved knowledge of mechanisms. As we learn more about microbiome, the players should be able to tell you how they're changing the microbiome. Even if we don't know the best microbiome yet, they should know how the microbiome is being changed and potential mechanisms of action for these products. We're moving, the big question today is, can you identify a mechanism of action, and therefore, is there a condition where that organism or where that product, no matter what it is, will be effective? We are in this one-size-fits-all category, but it's unlikely that one size does fit all. And if we know mechanisms, we can say which size fits when, or which product might be effective in a particular environment. Look for the suppliers to start taking samples from your facility, fecal samples, and start measuring fecal microbial populations. They'll want to gain some information in terms of what's present in your pigs, and they'll use that as well as they develop products and help to inform you how their product has changed the microbiome. As others have said, I think certainly the microbiome research has put our focus in sow and piglet management. That early life colonization pattern, we talked about humans the first 3 years. In pigs, you probably have the first couple of days in order to change something. And there are long-term, and we know there are long-term implications of early changes in microbiome for the pig as it matures. So managing the farrowing crate, managing the sow, is it possible to supplement core microbiota that support the pig later in life? Are we missing? Do we know? We don't know what the core microbiota are yet, but is there a single organism to be provided here or a cocktail of organisms that can normalize pigs in your herd and result in improved barrier function? This is just going back to the sow. I probably don't have time to do this, but This is a sow that's been fed soluble fiber in the gestation phase. It changes. These are 14-day-old pigs, and just bear with me, it does change the microbiota of the 14-day-old pig by feeding fiber to the sow. And these are a bunch of markers that indicate that these piglets at 14 days of age have an improved gut health. And in fact, they were heavier at 14 days of age, although I don't show the data. So focusing on the sow and the nutritional management of the sow, in terms of fiber composition, in terms of that transition from the gestation diet to the lactation diet, that's a significant disruption for the sow and probably having a significant effect on what she's shedding in fecal material, is likely going to be important with respect to early colonization in the piglet and probably needs some of our attention. We've talked about this, Emma's talked about this in the human society. Are we too clean in pigs? Are we doing too much sanitation? Have we kept pigs inside in sanitized environments for generation over generation over generation? Have we over time missed something? The classic example here is looking, for example, although we're all hating wild pigs right now in terms of their potential as an ASF reservoir, is there something in the microbiome of a wild natural pig that we've bred away in our domestic populations, in our commercial populations, that could provide some advantage? I don't think we know the answer, but it's certainly an interesting question. I will finish up very quickly, and we're sort of really pushing time here. This is a project that, and I'll just give you a brief overview. This is a project that Swine Innovation Porc is supporting to try to get a better handle on the microbiome. Our collaborators are here. It's a cross-Canada collaboration to try to get a better handle on what is the ideal pig gut microbiome. I'm not sure we'll get that specific answer for you. We do have a number of industry collaborators that are providing some of the leverage funding and folks in the industry that are working with us and giving us access to their animals and their barns, for which we're very appreciative. And what we're really doing here, I'm going to skip past this, is we're looking at sows. We are sampling vaginal flora and fecal flora from sows, and we're sampling piglets from birth through to slaughter, fecal samples. And we're hoping then, and we're doing that across Canada. We're doing that in herds in Quebec, Ontario, Manitoba, Saskatchewan, and Alberta. And from that, the principles that are driving the work is that we need to work on a commercial farm basis. One of the strategies or one of the concerns is that when we work in research facilities, they're too clean. Are we really seeing what's going on in a commercial setting? So we are going out into commercial barns and looking at commercial pigs. We have a pan-Canadian representation here, so we're seeing corn-fed and wheat-fed and barley-fed pigs, and we're looking at management and climate and diet variation within these populations. We want to maximize the number of pigs, and in order to counter that pig-to-pig and facility-to-facility variation that we know we're going to see. One of the big issues in microbiome, again, that dynamicism, is that no two pigs seem to be entirely the same. What is the core microbiome in the Canadian pig herd? And a focus on fecal sampling. Yes, we know that if we went to the small intestine or to the colon, we might get a more informative sample in terms of what that pig, of the relationship between the microbiome and the host, but we know we can't sample the colon or the small intestine as a diagnostic source in pigs, commercial pigs. So our only really relevant sample for diagnostic purposes is the fecal sample, and that's the sample we'll focus on. So we're doing studies in conventional and RWA farms. Again, we're going for sampling the sow, both her feces and her vaginal tract, and we're doing multiple collections from birth to slaughter, recording body weight, heart girth, health score, looking at the management strategies on each of these farms and trying to connect these fecal profiles in terms of microbiome with these phenotypic data that we can gather. There's about 6,000 samples that will be generated from this activity alone. We're also working with some of the genetic herds, nucleus herds. We would hope to sample at least 3 genetic lines, and again, go from birth to slaughter. The reason we're going in genetic herds, they're cleaner than most commercial herds, perhaps, in terms of the of management, but they collect feed intake and feed efficiency data on the male populations and some females, and that gives us a lot more phenotypic information to connect with microbiome. So we're looking forward to adding a sample set from that as well. Do core members vary with region? Do core members vary with growth rate? So again, basically these are questions about getting us to a place where we can say, these are organisms that you should promote, these are organisms that you should not promote. Maybe it's different in Ontario and corn versus the West on barley, but we'll hopefully start towards getting some of those answers. Ultimately, can we develop fecal diagnostic tools that allow us to tell you something about the fecal profile that you see in your organization, in your farm? And can we use that profile then and the information that we get with respect to the core microbiome in these pigs to help us develop new products? Good question. The earlier the better, I guess, would be my answer in that respect. You know, there are very few studies even at this point that have inoculated a pig at birth to see if what studies have been done is antibiotic studies, right? If you treat a baby pig with antibiotics, later in life you will see differences in the community in that pig. What we haven't seen, and maybe my colleagues know more, but where if you provide a probiotic or a group of organisms to a baby pig, does that change its community so that you can recover those again later in life, or does it dramatically change the community later in life? We haven't seen that yet. And so I think that's another experiment that still needs to be done. And then your question is, how late can you provide that new cocktail in order for it to be effective? Again, is the second part of the two-part question. So the question is, are we looking at a subset of pigs that are receiving zinc oxide or not zinc oxide in the RWA environment? And the answer is no, not specifically. In fact, the choice of RWA versus conventional was simply to meet that need for a variety of, or expectation for a variety of different production practices. And we're looking for, in fact, even in the samples that we collect, we're trying to differentiate, right, the high performers from the low performers and attempt to understand, can we differentiate the high and low performance based on the microbiota that we recover in the feces? So that's not a question that we asked specifically with respect to zinc oxide and looking at the RWA group. Renee?
Or even in the RWA?
Yeah, not in this study, and in fact, sorry, Renee's question was, are we in this study looking at pigs that might be housed outside? And in fact, specifically, we've avoided, for example, many of the organic strategies or organic production practices require pigs to be outside for a certain percentage of the day. Organic and RWA may be considered similar, but we've been trying to avoid organic so that we don't have pigs that are outside versus pigs that are largely maintained in commercial production systems, whether they be RWA or not. So that's something we specifically decided to avoid. Again, this is always about reflecting the diversity that's present in our commercial pig production, while at the same time not bringing in factors that we can't control.
So I'm going to ask a question regarding the poultry industry, and if you talk to, I heard a presentation by a poultry veterinarian from the University of Georgia, and they purposely have their houses on deep litter with broilers and bring in multiple crops without cleaning the litter out if they haven't had problems, with the idea of inoculating the next crop with the microbial population that's in the house. You have any thoughts on that?
Yeah, I've never seen that. I've never seen scientific data on that, but I've heard anecdotal data as well that that the third crop on the same litter will actually outperform the first crop simply because of the inoculating effect of a well-managed litter. I've never seen any data that has supported that, but it's certainly anecdotal.
Yeah, the idea on that is that the litter over time will evolve microbially. And so birds within a few days, 14 days, get really good, solid establishment of microbiome that they
Exactly, and right, poultry is a unique situation, right? They're hatched in a completely controlled, sterile environment, and that early inoculation.
hatcheries are pretty dirty sometimes. So going up to the breeder hen, whatever you treat with her, antibiotically, that can have a transfer into the chick, nutritionally as well.
I think it depends on what you call really dirty or not versus a piglet coming out of the back end of a sow in a farrowing crate versus an egg in a hatchery.
They've had lots of issues with chick quality out in the field.
Sure.
Infections and poor performance. So they're dealing with it.
Yeah.
They don't definitely have 3 years to work to establish their microbiome. Any last questions?
Denise?
Can you discuss whether you predict you'll actually find core members for an indication of overall diversity? Which do you think is really big? Like tools
The question is,
what tool to quantify diversity?
quantifying diversity isn't that tough in terms of, we'll sequence everything and we will be able to assess the diversity that we see in all the environments that we're testing and with age. So we will certainly be able to report on diversity. Reporting on core members and being able to link core membership with outcomes, the phenotypic outcomes that we're able to connect with those microbiomes is going to be a much more difficult question. I don't know the answer to it yet. That's why we're doing the study. I think we'll see different core on certainly a corn, soy versus a wheat, barley, canola, right? It would be very interesting is that, what is common between those two extremes, and is that commonality and in any way associated with some of the performance outcomes that we can measure.
Final question.
Just on your
Right. So the question was relating the work that we're doing with microbiome sampling with respect to the age at weaning, and so the answer is we recognize there are various ages at weaning across the Canadian herd, and we consider weaning the great equalizer. So we're taking samples at 4 days of age, simply because 4 days of age is a time we can't be there at farrowing. We don't want to disrupt farrowing, certainly not in a commercial operation. So 4 days of age is our compromise in terms of an early sample. We're taking something mid-lactation. We're taking a sample 1 day before weaning, whatever your weaning day is, and certainly we'll record that, and we'll be able to try to link weaning day with, or weaning age with microbial outcomes. After that, we go weaning plus 1 week, weaning plus 10 weeks, weaning plus 17 weeks. So weaning is, we have birth and weaning in our equalizers in assessing the longitudinal changes.
Okay. Thank you again. Let's express our appreciation to Andrew.